Dr. Stephen A. Rosenberg is known as the father of cancer immunotherapy. His career at the National Cancer Institute has been defined by one radical idea. The best defense against cancer isn't chemotherapy or radiation, but the patient's own immune system. This belief was born during his surgical residency. In 1968, he operated on a patient diagnosed 12 years prior with inoperable metastatic stomach cancer. To his astonishment, Rosenberg found no trace of the disease, spontaneous regression. A second case confirmed his hypothesis, a cancer patient on anti-rejection drugs. When the drugs were stopped, the cancer vanished. These observations, which he details in his book The Transformed Cell, led Rosenberg to a profound conclusion. If we can spark a strong enough immune reaction, we can make even the most established cancers disappear. This set him on a 50-year path to prove that the body could rid itself of cancer if properly directed. Rosenberg's first major maneuver focused on interleukin-2, or IL-2. This protein acts as a growth factor for T cells, the body's immune soldiers. Early adoptive cell transfer studies used IL-2 to activate non-specific peripheral blood cells, resulting in lymphokine-activated killer, LAK cells. Based on animal data, Rosenberg developed a high-dose IL-2 regimen. After a grueling initial series of 66 clinical trials in patients with metastatic cancer yielded no successful outcomes, the 67th patient, treated in 1984 with an aggressive regimen of high-dose IL-2, experienced a complete and durable cancer regression. This landmark event unequivocally demonstrated that a purely immunological approach could mediate the complete destruction of large, established cancers in humans. This success, which led to the FDA approval of HD-IL-2 in 1992, came despite severe dose-dependent toxicity requiring highly specialized critical care. This inherent limitation drove the subsequent research to develop a more focused, targeted approach. The toxicity of systemic IL-2 demonstrated that the strategy needed to be more targeted. The transition from LAK cells to tumor-infiltrating lymphocytes, or TILs, marked a critical evolution toward precision in ACT. Rosenberg's team began culturing single-cell suspensions derived directly from resected solid tumors. These cultures yielded pure populations of lymphocytes that had already infiltrated the tumor in vivo. These TILs, unlike LAK cells, often exhibited specific anti-tumor reactivity against the tumor of origin. This groundbreaking finding provided the first direct clinical evidence of the effective role of tumor-specific T lymphocytes in human cancer immunotherapy. The ability to grow large numbers of these specific anti-cancer T cells in vitro using IL-2 immediately established TILs as a highly promising therapeutic platform. The TIL therapy protocol is a sophisticated form of adoptive cell transfer. It begins with the surgical resection of a tumor. The TILs are isolated and then expanded ex vivo in high concentrations of IL-2 to generate billions of tumor-specific cells. Before reinfusion, patients undergo a preparative chemotherapy regimen known as lymphodepletion. This is a vital step designed to temporarily deplete endogenous immunosuppressive cells and create an immunological vacuum that maximizes the persistence and proliferation of the infused cells. Following the reinfusion of the expanded autologous TILs, high-dose IL-2 is administered to ensure the cell's survival and exponential proliferation in vivo. This dual requirement, lymphodepletion followed by high-dose cytokine support, demonstrated that merely identifying the correct cell population was insufficient. The host environment had to be rigorously prepared. Optimized TL protocols have achieved objective response rates up to 50% in metastatic melanoma, often leading to long-term survival, establishing TILs as a distinct and highly potent therapeutic modality. Rosenberg continues to drive research to apply TIL therapy to traditionally cold, solid tumors like breast and gastrointestinal cancers. The success of TILs was the launchpad for genetic engineering. Dr. Rosenberg extended the foundation of AC by pioneering the field of human gene therapy. In 1989, he achieved the historic milestone of being the first researcher to successfully insert foreign genes into human beings. This initial trial, designed to track the fate of infused cells, successfully established the safety and mechanism for retroviral gene transduction in humans. It launched the entire clinical development pathway for engineered cell therapies, leading directly to the modern revolutions of chimeric antigen receptor, CAR, T cells for blood cancers, and crucially, T cell receptor, TCR, therapies for solid tumors. Rosenberg's contemporary research focuses on high-precision targeting. A defining triumph was documented in 2016 against mutant KRAS G12D, one of the most intractable oncogenes prevalent in pancreatic and colon cancers. Rosenberg and colleagues identified a polyclonal T-cell response against the mutant KRAS G12D neoantigen in TILs. A patient infused with these specific TILs experienced objective regression of all targeted lung metastases. This case provided concrete evidence that ACT could successfully target driver oncogenic mutations in common solid tumors, validating the pursuit of highly personalized neoantigen-specific therapies. Dr. Stephen A. Rosenberg's lifework has permanently transformed oncology. He established immunotherapy as the indispensable fourth major treatment modality alongside surgery, radiation, and chemotherapy. His progression from the non-specific high-toxicity IL-2 regimen to the tumor-specific TLs, and finally to the precision targeting of neoantigens, illustrates the constant, iterative refinement necessary to optimize the therapeutic index of ACT. His legacy is defined by his commitment to translating radical scientific hypotheses into life-saving treatments.